Hello, everybody. Welcome. We're going to just give it another minute or two and get going.
How about we get started? And Monica can always join in later. She's our second speaker. So, and if at worst, that she can go last. I don't think it's a big deal. All right, everyone. Well, welcome to today's talk, which is entitled The Best of ACMG 2026. This is our first edition of covering the most important and most impactful sessions from this year's ACMG. I'm very excited to present the speakers today. We have Alexa Dixon, who's a senior clinical variant scientist at Washington University. We also have Monica Bowen, who is a variant assessment scientist too at Ambry Genetics, and Matthias Wilkie, who is a clinical variant analyst also at Washington University. So today our esteemed speakers will be highlighting the key takeaways from this year's ACMG meeting that are relevant for variant scientists. So we just want to go over very quickly today's agenda. So Alexa will be highlighting the workshop session on Tuesday entitled Beyond Uncertainty, using functional assays for clinical variant classification. Then Monica will give us some key takeaways from the concurrent session on Wednesday entitled Dilemmas of Omic Technologies for Molecular Diagnosis of Rare Disease, and also be offering some poster highlights and then talking about the Variant Scientists Network Ancillary event. And then finally, Matthias will share information about a useful tool discussed during the plenary session, as well as key takeaways from Wednesday's Platform A titled Laboratory Variant Classification. Then after their, just want to change slides. After their talks, we'll go into a question and answer period. And I wanted to let you know that the talk will be recorded and posted on the Variant Scientists Network website at a later date. And of course, if you want to learn more, the ACMG 2026 sessions are published in the open access journal called Genetics in Medicine Open. So I'll stop sharing and you can take it away, Alexa.
Great. Thanks for that introduction, Yasmin. I'm going to share my screen and get started. Apologies, technical difficulties. Again, you know, as long as we've been in this pandemic, you would think we'd have this all put together. All right. I assume you're seeing the right slide. So I'm going to be talking about the
Yes, I'll go
workshop
check.
event. Thank you. I'm going to be talking about the workshop event from Friday. This was a 4-hour workshop that I'm going to try to summarize into 15 minutes. So I'm going to be leaving out a lot of details. If you want to learn more, a lot of these slides were shared with me through the Atlas of Variant Effects. I highly recommend going to that website, Atlas of Variant Effects, and learning more about this. So the entire session was about the assays called MAVES, and MAVES stands for Multiplex Assays of Variant Effect. And these are functional assays designed to determine effects of variants on the function of proteins. What you do is you select some sort of a cell culture model system. You then create a library of essentially any variant that could occur in a particular protein. You transfect those libraries into your cell culture model of choice. You apply some sort of a selection that would render some variants no longer able to produce their function and potentially kill the cells. And so you can see some of the variants here are creating a loss of function essentially and killing the cells. And then you sequence to read the results. This would then give you the wild type count versus any variant count, which you can then plot by every amino acid on the y-axis here. So this would be like methionine, serine, threonine, etc. And on the x-axis is the position in the protein itself. So position 1, 2, 3, 4, etc. And then you can plot the effect, either if it behaves in a positive way or more like wild type, or if it behaves in a negative way or is deficient. And then once you plot these scores, then you can use a data set of known pathogenic or known benign variants to be able to determine at what strength then can you take this functional evidence and turn it into evidence for classification. So in this far left graph, you can see that the known pathogenic and likely pathogenic variants are segregating very well and separate from the known benign variants here on the right-hand side in blue. And because these two populations are separate from each other with very few overlapping, the calibration for that level of evidence would be strong. Here in this middle graph, you can see that there is some overlap between the known benign and the known pathogenic variants. And so in this instance where they're a little bit more closer together and there's some overlap based on a calibration technique, you can apply these level of evidence at the moderate strength. And then on the right-hand graph, this is a different graph showing, in this instance, the known benign and the known pathogenic variants are not very well separated. And so you would be able to apply any assay output from this particular assay at the supporting level of evidence. So this would require, of course, a particular gene that the function of the gene is known, and luckily we have a lot of those available. One of the best known genes for working with MAVES like this is the BRCA gene. So this is the depiction of the various known functions of BRCA. So there's molecular interactions that we know, and through assays called VAMP-seq, which is variant abundance by massively parallel sequencing, you can look at these particular interactions. One of the major functions of BRCA genes is to function in DNA repair. So you could take one of these assays and look at the major functions. And then for saturation genome editing, kind of putting it all together and looking at the cell fitness would be saturation genome editing. So there's all of this data out here. There's lots of basic science going on, producing MAVES in with different genes in different assay systems. But there really wasn't a way to pull all of the data together and make it easily accessible. So This group created this MAVE data repository at mavdb.org, and this is a screenshot of mavdb.org. There's a bunch of assays that have been deposited into mavdb.org, but it still wasn't easily accessible for people who are not very familiar with the MAVE technique. People like me, who's more of a variant scientist, I want to know, what is the impact of this variant on variant classification? How should I incorporate this functional data into the variant classification and make it easily accessible for providers who want to explain the information to their patient? So they took this database a step further and created the MAVES for Medicine, or MAVE MD, and this is the website for MAVE MD. They've got a bunch of genes that they list that have information in them. And if you were to click on one of these genes down here, so for instance, if you were to click on BRCA1, it would bring you to the functional assay page. I know this says anatomy of a variant page, but really it's the functional assay page. And it gives you all kinds of information on this particular functional assay. So here at the top, you see those score histograms, those score intervals that I showed you earlier. Then it gives you information about how they calibrated, or how these assays would be calibrated, and that ACMG strength style evidence that I showed you, whether it be strong or moderate or supporting. It also shows you the heat map that I mentioned earlier that gives you all of the potential amino acids on the y-axis, and then the position of each amino acid in that protein, and then the color of the dot refers to the output of the assay itself. And I'm already running out of time, but there's extra information there. They walked us through a particular pedigree example for BRCA1. Essentially, on initial look, there wasn't enough information to call this anything but a variant of uncertain significance based just on absence in population databases and the prediction scores. However, there was a MAVE that had been deposited into ClinVar. And if you use the MAVE MD database, you can see exactly where the patient variant falls within these altered functional score histograms that I showed you earlier. It even gives you a nice summary that it's functionally abnormal, and at what strength can you apply the functional score. This one's PS3 at the strong level, and based on that, they were able to reclassify this variant as likely pathogenic. So 4-hour session summarized down into 15 minutes. MAVE's strengthen variant interpretation with functional evidence. It improves classification confidence. How confident are we that this variant is what we think it is? We're confident now that it's likely pathogenic based on that functional evidence, and it bridges the gap between research and clinical practice. And with that, I'll hand it over, I think, to Matthias, because Monica is,
Oh,
I
I'm
think,
here.
still. You're here.
I
Oh,
joined.
yay. Fantastic. Great.
Thanks. Second. Okay. Hello, everyone. Thank you for joining today. My name is Monica Bowen, and I'll be covering omics networking and posters at ACMG. As a reminder, you can always view the full program using the QR code on the ACMG website, and you can search for abstracts by poster number or author name there as well. So the first session I'm going to cover is the Wednesday scientific concurrent session on omic dilemmas for molecular diagnosis of rare disease. This was a 5-speaker session that focused on just a general introduction to different types of omic technologies, sharing of examples where long-read whole genome sequencing and epigenomic testing could provide additional insights. And then there was a session on transcriptome sequencing by Baylor College of Medicine. And then the last 2 sessions focused on ethical challenges in returning results using omic technologies. And I think this session was pretty successful in meeting its objectives, which were to identify the factors that influence the return of omics results to rare disease participants and identifying strategies that can enhance their clinical translation. And then they also focused on describing what it looks like to implement new omic technologies using the example of transcriptomics in a CLIA-certified laboratory. And then last but not least, again, exploring some of the ethical principles and how those might impact our work as variant scientists and also the work of genetic counselors. So to start, omics, when we say omics, we're distinguishing genomics from other types of omics, whether that be the new genomics of long read genome sequencing, epigenomics, or transcriptomics, or sometimes metabolomics or proteomics, right? And so what they underscored in the primary session was that genomics is often thought of as this binary static thing, right? It's our genetic sequence, whereas transcriptomic data is, first of all, continuous, right? You don't just have one or two changes at a base of your DNA. You can also have active changes in response to your environment. So that poses a significant challenge, but also provides additional insight when looking at context specificity in disease. So some of the examples that they shared were the advantage of long read sequencing for identifying expansions in a particular epilepsy gene that was missed by short read sequencing. However, they face some challenges in reporting this particular expansion because there's not really reliable population data for some of these. They also showed examples where epigenomics and methylation signatures could distinguish SOTOS patients from controls. And then the Baylor talk shared some of their findings using fibroblast versus blood for RNA-seq studies and how to weigh data where you don't have apparent splicing detected or attributed to a specific variant necessarily, but you do see things like that particular gene is an expression outlier when you look at the data as a whole. And so in that particular case, they decided to use that data when reporting back to patients using the tagline investigational results. They also polled the audience on whether they would want to report and or receive that type of information themselves. And it was dead split across the entire room. Some people thought more data is good. Other people thought there was no use for it in the absence of other guidelines. So then we move on to the ethical considerations for this session. Chief among them were questions of data transparency, result actionability, and patient autonomy. That often drove why patients wanted access to this data, but also underscored why scientists and clinicians were hesitant to provide it, because there are concerns around adequate staffing and funding to maintain the guidance required to make those results translatable as complexity increases once we adopt additional technologies. The other take-home message was that expanded omics equals an increase in incidental and secondary findings, which adds to the burden of not only of variant interpretation, but also genetic counseling. A particular genetic counselor from the NIH shared a fraught genetic counseling experience she had with a proband whose whole genome sequencing and long-read sequencing revealed dual diagnoses that had implications for the family members, but due to personal and religious reasons, it was very, very challenging to navigate that process. And so, again, the insights are that we are in a process, in a place right now where we're transitioning from using omics in a research setting to a clinical setting. This can be a challenge in terms of deciding when it's worth reporting, but it can shed additional light, especially when you're stuck at a variant of uncertain significance classification. These technologies are rapidly changing,